Hello metric students, welcome to our next lecture on experiments, chapter 10 from real econometrics. Today the main thing that we're going to talk about is balance. So if you remember, we were talking about experiments and randomization, and I mentioned that it's the gold standard here of establishing causality. But what we're talking about mainly in this chapter is the fact that it's not foolproof, and in particular, how it can go wrong. But what I want to revisit in order to emphasize how it's not foolproof is going back to this causal model, which I mentioned in our first lecture, and I mentioned very on in the semester. But the true average effect of a treatment, in thinking about the truth that's out there, is actually how does it impact an individual. And we can't see both those two states of the world where the individual has the treatment and the individual does not. So our notation that we've used notes that the treated individual is that given sign, the treatment equals 1, as for individuals. The subscript on the y is telling us we're looking at the state of the world where the treatment is received. So on the left-hand side, this is an observable thing where the individual who is one of the treated individuals, we're looking at the true, actual, observable state of the world where the treatment is received. Now, if we really wanted to get the true average treatment effect, we would see a different state of the world, but for that same treated individuals or that same set of individuals. We would see the state of the world where the treatment was not received. But instead, what we see is the observable treatment effect, where we're still looking at the state of the world where treatment is received for the treated individuals, but we need a control group. We need a different set of individuals that did not receive the treatment, right? The non-treated individuals. That's the difference between the observable and the true value. So the necessary condition for us to be able to capture that true value with what we're observing is that the treated individuals have to be well approximated for by those non-treated individuals. So these are the conditions, and what I also want to emphasize, this is the one condition, is that this, you may not have put it together in your head, but it's essentially the same thing as causality, as exogeneity. You may have not completely put this in your head, but it's the same idea as exogeneity. We have the necessary condition is that the treatment groups and control group in the state of the world that neither of them received the treatment look the same. What we're saying with their exogeneity condition is that all the other factors that influence our outcome aren't related to X. In other words, they have to be the same for the treatment and control. Here we have states of the world for the Rubin's condition. For the exogeneity condition, we have regardless of the value of X. That the average outcome is equal, or the other stuff that influences the average outcome is the same. These are the same ideas. With randomization, we're randomizing who is treated and who gets the control. Therefore, unless something is terribly wrong, and we'll talk about how we look at that, unless something has gone wrong, we would expect that treatment shouldn't be correlated with anything. It's randomly assigned. And so we'd expect that the Rubin condition holds, exogeneity holds. Right? So randomization gives us those conditions that are necessary for establishing causality. Now let's finally talk about balance. Balance simply refers to the idea that after randomization has done, where we've put some people into a control group, other people we've decided to treat, after we've done that randomization, we can look at the observable other x's or observable anything and say, does our treatment group look like our control group? That's what we're talking about when we're talking about balance. So the way to check that is to simply look at the difference in other variables. We can do a t-test. We can look at differences in means. We can do an OLS regression and look at the p-values, which is the same idea. And the hope here is that if we're looking at all the different observable values and whether or not we're balanced on those, that if we're balanced on the observable values, then hopefully and probably we're going to be balanced on those unobservable values that we can't control for. Because we know how to deal with different x's. That's not necessarily a problem. What we can't deal with is different error terms for treatment and control. Let's talk about things that cause our sample and samples in generals to be unbalanced. Sample size is a big factor. The smaller your sample size, the less likely you are to reach that balance. There's simply more noise. This actually brings us back to another early topic in our course, the variance of the average formula. Because what we're doing here is we're comparing averages, the average for the treatment and the average for the control group. And we're asking, are they equal? Well, the question is going to depend on whether we answer this is very dependent on the variance. Right? If you have a lot of variance, then maybe they're not equal, but because of random noise, because we don't have a large sample. Adding more and more observations lowers the variance in a very predictable way, as you see this formula right here. Another cause of an unbalanced sample can actually be poor sampling methodology. Perhaps there is a selection problem that you didn't consider when you were constructing your experiment. Maybe, well, we talked about the issue of what happens when there's non-compliance. Perhaps you're not paying attention to that, or perhaps there's a selection issue in terms of the treatment and control. That can lead to an unbiased. You also could just have sort of bad luck. You could have a random chance that drives this. Now, when we're checking for balance, the thing that we need to remember is that with a 95% confidence level, that means that 1 out of 20 t-test will spuriously show that the difference between the averages is significant. So it's not the end of the world. You're not sunk if you're doing your diagnostics for a balance and you're showing some of your covariates are unbalanced. So the ways to deal with them, I'm going to break, this is going to be very general, high level. We're not going to go into much depth here, but we can think about the ways to deal with them before you are putting together your sample and ways to deal with it after. So these are preventative measures before you have run or even assigned treatment and control in your experiment. You want to think very long and very hard about the potential for selection. Could your subjects, however the mechanism works, be able to decide whether they're receiving the treatment? Could there be any selection going on accidentally by the way that you're constructing your sample? So think very long and hard about that potential. You also can assign people into treatment and not treatment groups based on the covariates. So you're going to randomly assign them, however, you're going to select a certain number that have each particular characteristic. So maybe you're looking at a group that includes both professors and students, and you want to make sure that it's balanced, that there are equal in each. So you take your professors and you would say, all right, we're going to randomize and do 50-50 amongst your professors. You would randomize amongst the students, 50% of each, and that way you're knowing your treatment and control group are balanced along that dimension. This is called blocking for people who do statistical sampling. This is an important technique. There's a lot of literature and reading you can do about doing this. The one drawback it has is as you divide more and more groups, the number of people you need in your sample to be able to say anything very rapidly becomes prohibitively large. So if you're doing professor versus student, male versus female, if you add racial categories, if you add other covariates, you rapidly are needing to have a really large group for every single one of those cells, which can be quite small and prohibitive. The other way to deal with an unbalanced sample, or to prevent it, is to make sure that you can get a big enough sample. Sample size is a very important factor in achieving balance in experiments, and it's very important to have that if you're going to be able to do your statistical test appropriately. Post-experiment, once you have your data and you are looking at your outcomes, what we can do is, when we see something that doesn't look balanced, we can control for it. It's actually not an issue to have an unbalanced sample if it's only unbalanced on the things we can observe. We know how to do that. We know how to do multivariate OLS. That takes care of that. The difficulty is, well, if you're unbalanced in something we can see, the honest truth is you might be unbalanced as well in something that you cannot. And there is no sort of easy way to deal with when the error term is endogenous. We've talked about some techniques, but there's no easy way to get around and escape that in this particular example. I hope you all are doing well, and I look forward to interacting with you soon. Take care.